morning. As Gunnar said, my name is Ólöf Dóra Bartel Jónsdóttir, and I am a senior scientist at Aquaplaniva in Norway. And this this presentation was made both of myself and Albert, as Gunnar said, Albert was supposed to be here. So the first half of this presentation is Albert's field, and the second half is my field. So if you have deep questions in the first half, then contact Albert. And my closest co-workers in Norway are Dr. Simo Madonna and Snorri Hagen, and they work at Nibio in Norway, and also Patrick Reynolds at GIFAS. I've also worked with excellent people at Matis in one project, and yes, and they'll probably be here, I'm not sure. Today I'm going to present our project on lumpfish, where we focus on lumpfish as a cleaner fish. There are several problems concerning lumpfish as a cleaner fish, and we have to look at them, of course. The main problem has been that only part of lumpfish, which is together with salmon in sea pens, are grazing on lice. The figure with the red bars show that in the first days of being with salmon, only 10% of lumpfish are eating sea lice or lice. But that number increases slowly but surely up to 36% as the days passes. But the grazing seems to stop at around 40%. And we would much like to have that proportion of lumpfish eating lice much higher. We have seen that there is a large individual differences in lice grazing of lumpfish. You can see that in this figure at the bottom. Some individuals hardly eat lice whatsoever, but others have more appetite for lice. That is, for us, very important and give us the reason to look at these individual differences specifically and raise the question if it is possible to select for lice grazing. At GIFAS there has been performed an experiment to see if it's possible to teach lumpfish to eat lice. And it has been concluded that it is possible up to some degree, using frozen crustaceans. And that would be important to stimulate the appetite for lice. Our project on the lumpfish is called Select and is ongoing. In this project we focus on family production of lumpfish, field test of course, data analysis, and the genomics. And that's where I come in, and my co-workers at Nibio. Our industry partners are first and foremost Leroy, but also Lumarine and Nordic Opdrett. As I mentioned earlier, the proportion of lumpfish eating sea lice are up to 42%. And keep in mind that some individuals not have the appetite for lice whatsoever. And therefore the lice grazing in some lumpfish groups can be very low. The fact that lice grazing by lumpfish does not exceed 42% means that for every 100 Norwegian kroners that goes into buying of lumpfish, a maximum of 40 kroners is used for delousing. And we want to get that percentage of sea lice grazing much higher, or up to 40 or 70 or 80%, to increase the sales value of the lumpfish fry produced. In this project we focus on how we can increase robustness and survival of lumpfish in sea pens, and how and if it is possible to increase the proportion of lice grazing among lumpfish. And we estimate that the value of the implementation of the project for industrial partners to be around 5 to 10 million kroners annually per 1 million fry under the first and second generation of the breeding project. The motive for our interest in looking closely at families of lumpfish in sea pens with salmon was the results we obtained 6 years ago. GIFAS performed a family experiment where siblings of lumpfish were together in sea pens and found that in 2 sea pens, you can see the arrow pointing at those particular sea pens, it was less infected by sea lice than salmon in other sea pens. In fact, there was a huge difference. In those 2 cages or sea pens, the reduction of sea lice on salmon was more than 80% compared to other. Those results suggest that there is significant maternal and paternal effect. Thus, the selection of optimal lumpfish was possible. It is possible. Selection focusing not only on lice grazing, but other important traits too, such as survival and robustness. In the autumn of 2018, another family experiment was done at GIFAS using 5 males and 10 females, producing 10 families. You can see those here. This is the families. The result indicated a clear parental effect on offspring when it comes to lice grazing, among other traits. And as you can see in this figure, the salmon in pen 207, you can see the brown or the red arrows pointing at it, had the lowest infection of lice, and in this sea pen, 2 half-sibling groups were present. Half-sibling having the same father but different mothers. The research has been published in those 2 papers, one in 2016 and one this year. And our trial showed that it is possible to significantly increase lice grazing through genetic selection. We also see clear effect in disease resistance and survival. And now I will move to the genomic project of the genomic work of the project. What we're doing can be divided into 3 categories. I will call it A, B, and C in the other slides. A, population genetics, where we use wild samples from different locations to analyze population structure of lumpfish. This is done both on small geographic scales, such as along the Norwegian coast, and on large geographic scales, such as the Atlantic. We use 2 different genetic markers. One is called microsatellites, and one is called SNPs, and stands for single nucleotide polymorphism. And microsatellites can be divided in 2 groups. I'll be talking about that a little bit later. Okay, B category. We do genome-wide association tests, marker by marker, and locate each loci into 25 chromosomes that lumpfish has in its cells. We also map the annotation of microsatellites on active region of genes according to their biological function. And the C category, we aim to develop genome-wide linkage map and link together specific traits to genotypes. In that way, we will be able to point out a specific genotype that is linked to favorable traits such as lice grazing, survival, and robustness of lumpfish. And first, the A category. I'll tell you a little bit about the population genetics. This is the sample size of wild lumpfish that we have been using in this project. We have analyzed the population structure along the Norwegian coast using both microsatellites, 2 types, and SNPs. And when it comes to the North Atlantic, we use only SNPs as a gene marker. And as you can see, we have samples from both the west side of the Atlantic as well as the east. I'll tell you a little bit about the genomics in the Atlantic. We use, as I said, SNPs to map the population structure of lumpfish in the Atlantic. The figure on the right, the smaller figure, is a cluster analysis showing clustering of samples into groups based on genetic similarities detected by SNPs. Different color indicate genetic differences. Within each plot, vertical bars that you can see at the bottom, vertical bars represent individuals, while colors indicate a different cluster detected. The results from the Atlantic show clear population differentiation from west to east. You can see that Norway, Iceland, Faroe Island, and Ireland are looked at as one population, whereas England is a different population, and we have a total different population in the west from Greenland and Canada. And you can also see it in this, we use different colors. This is one population, this is another, and the third, according to SNPs. And when it comes to the smaller geographic scale, we looked at the population structure along the Norwegian coast using microsatellites. And as I said, we have 2 types of microsatellites. One is called GSTR, and that's a microsatellite located on inactive region of genes. And then we have another type of microsatellites called EST, STR, and those microsatellites are located on active sites, active regions of genes. So, we have done analysis with both. And in both cases, we do not detect any population structure of lumpfish along the Norwegian coast. And it's best to look at the cluster plot here in the middle. Each vertical line bar is individual, and as you see, we just have 2 colors indicating that we have one population of lumpfish in Norwegian water. However, we have also analyzed samples from the Norwegian coast using another genetic marker, the SNPs, and another samples. And we are seeing some kind of genetic structure between south and north, but this we will have to look into deeper. We're just starting to analyze with the SNPs. Okay, the B category. This is a figure from the B category. We use recombination rate, also rate of crossing over of genes, to find out the position of genes on the chromosomes. Lumpfish has 25 chromosomes, and the location of genes that has been discovered are now defined on the chromosomes. And category C, we are currently working on connecting interesting traits to genotypes. It's a method called QTL mapping, where QTL stands for quantitative trait loci. We are particularly interested in finding a relationship between traits related to growth, body weight and sustainable growth rate, health, such as cataract severity and other things, and lice grazing, of course. So we have 4 key phases in molecular breeding, and they are phase 1, the development of molecular markers. We're done with that. That's the microsatellites, 2 types, and SNPs. Linkage mapping, that's defining position of known genes to the chromosomes. The analysis is finished, and we are in and the publications are in process. Quantitative trait loci and genome-wide association study analysis. This phase is in process, and yes, we're working hard on it. And we're yet to reach the phase of marker-assisted selection breeding. This is what we have published so far. 2 publications on the matter. One is Population Genetics, published in 2018, and one on genome and transcriptome analysis, published this year. And there are many more publications in process. Thank you. Any questions? Yes.
Thank you very much for a very interesting talk. I have one question. If already now could see, if you could see any association with the inheritance of the traits for growth, health, and lice grazing.
We do. We do see a clear linkage between genotypes and those traits, but we have to analyze it such as we can do it to special genotypes. So we have 1 trait and 1 genotype, or more genotypes we have to see. So that's what we're working on, but there are connections.
So are the traits located at the same chromosome? Are they
No.
Okay, so you have to do a lot of breeding to
Yes.
I have one question.
Yeah.
It seems to be working using Lumpfish to eat the sea lices and salmon lices, but the environmentalists are talking about it as a disposal fish. Does this worry you, or is that something we should consider, or try to find something useful to do with it after we have, because they're not eating lice after that?
Yes, absolutely. Yes, there has been ongoing project to find a way to use the lumpfish after they've grown too big to eat sea lice. But to my knowledge, we don't know actually what to do with it. But it's not my special field.
No, I know.
Yeah. So many of you have probably better answer.
When you grow these fish, they don't eat sea lice.
When they're small, they eat sea lice, and some eat more than others. But when they grow bigger, they stop eating sea lice and start eating pellets.
Excuse me, when I was I lived in Norway before, and I saw that we were introducing these tunnels that the fish could swim through and just take off the lice without any chemical or other things. Is that? Have you heard of it? And do you know?
I've heard of it, but I don't know much about it. I'm sorry, that's not my field. It's just the genetics, that's my field, actually. Yes?
How big data set do you have behind
It's called cleaning stations, and they are doing some actually research on that in North Norway. Yeah, seemed to work, that the fish was swimming into that, which is known also in nature. Yeah. And then it was also a shelter for the lumpfish. But for some reason that has not really been taken up. So I don't know what is the end result.
There was another question?
Yeah, how big is your data set behind GWAS analysis? How many samples of phenotype?
We have, now we have around 300 samples behind that. Any more questions? Thank you.